they measured the clinical research outcomes in 2012 and they published some guidelines. While whenever physicians are prescribing trimethoprim, it should not be prescribed in neurodegenerative patients, particularly Parkinson's or movement disorder patients. They should not prescribe this drug. And one more information, and renal impairment patients. So based on the severity, GFR, EGFR, which we need to consider mild, moderate, severe, and if it is severe, this drug should not be given. And if it is moderate, the dosage must be reduced. Usually they used to go for BD. The challenge, because why I am talking this, still we are facing the challenge. And see, if you are telling sir the dose must be reduced, some physicians, they are ready to accept and they've changed the prescription in our hospital and they made it once daily. But other physicians, they are telling no. If you are managing this condition, automatically it will be alright. They are not ready to even if you are showing the evidence. So there is a need for evidence-based information for each and everything. So that's why once the clinical research is over, sponsor is very particular about their therapeutic outcome. They are not considering the other people's. Like for example, antihypertensive drug which comes to market, the sponsor focus is only on whether the drug is having antihypertensive effect or not. They least bother about whether the patient is obese or patient is having comorbid conditions. No, their primary objective is whether this drug is having antihypertensive activity or not. Right? So coming to this is the reality. So whenever we are coming to conclusion, any of research, so based on, see for example, I just want to give one more example for this, apnea patients, right, sleep apnea patients. So it was proven that a moderate sleep apnea patients can be well managed by CPAP therapy. In case, if patient is obese, in case, if patient is with diabetes, in case, if patient with comorbid conditions like cardiovascular complications. So what is the outcome? It's already proven, NCPAP therapy is effective in moderate apnea patients. But what about the comorbid conditions? Whatever the research which comes out every day, we can see in the newspapers, every day a cup of coffee, right, will keep you away from these things. But it's based on the limited number of population, but in our case we need evidence-based information. So coming to the next slide, why clinical research, outcome research? Clinical outcome research and pharmacy practice is essential to sustain the growth of maturation of profession in the evolving healthcare system. So we need to work on clinical research outcome measures. Coming to the measures, outcome measures. It's really interesting while we are talking about measures. Of course, only 50 years are familiar with outcome measures, measures of occurrence and measures of incidence, all these things which you are familiar. But the same thing, theoretically you people are strong while you are going for your research. What you are going to measure? What are the variables which you are going to collect? If you are asking something, and again, there are some research works, they used to start without having any primary object or research question. They used to start, and after that, whatever the data which is available, I am going to collect. I will compile all these things at the end of my work. What is the variable which I am going to collect? In what way I am going to apply that variable? What exactly I am looking from that variable? No one knows. While we are starting a research, simply we used to go and we will collect the data, and we will, after that, whatever the data which is available, according to that I will perform statistical analysis. This is what the reality, right? So, outcome measure are the tests that investigator performs to decide whether the treatment being tested in the clinical trial is having any effect, which is called as clinical research outcome, right? Measure. So the selection of appropriate outcomes or domains is critical when designing clinical trial in order to compare directly the effect of different interventions in the ways that minimize the bias. There is a growing recognition that that insufficient attention has been paid to outcome measured in clinical trials, because their primary objective is to find out the clinical outcome, whether from the sponsor side they are very particular about their clinical outcome. So outcome research methods, all these contents are available in our textbooks, right? I'm copying and I'm just giving in front of you. So everyone knows it while we are talking about clinical trials, two categories, interventional and non-interventional. Coming to the non-interventional, observational studies, which is nothing but cross-sectional studies. Coming to the outcome, echo model, everyone knows it. So the three things, one is economic, and another one is clinical, and another one is humanistic. So these three outcome measures are widely used. Whatever the study design which you are going for clinical trial or clinical research, either it may comes under economic or clinical or humanistic. So whatever the outcome which you want to measure, these are the methods. Either you can go as a retrospective chart review and prospective clinical trials, meta-analysis, observational studies, and computer modeling studies. So I'm completing the first part of clinical research outcome, and clinical research outcome measures. So now I'm going to talk about meta-analysis. So why meta-analysis? Is it mandatory? Again, while talking about meta-analysis, many people, their opinion is different regarding meta-analysis. Some people are telling that meta-analysis always favors sponsors, because whenever they are performing meta-analysis, they used to go with the selected number of articles which favors the sponsor, what sponsor is expecting. So that's why many people are telling meta-analysis, no, we won't believe meta-analysis. But the thing is, here we are not going for that controversy whether meta-analysis are good or not. Anyway, it was given in our syllabus, we need to study what is meta-analysis. So is it possible to carry out the study in meta-analysis? This is what actually we need to learn. So coming to the evidence-based information. So whether meta-analysis will provide the evidence-based information. So here we can see three different patients. If you are taking a treatment guidelines, any treatment guidelines, so there will be a first choice of drug, will be one drug. Okay. So, but if you are going to the individual patient expectation is different. Some people may go with lifestyle modification, some people in simple we can say it's analysis of analysis. What is analysis of analysis again? So here it's 100% table work, right? 100% table work, collecting some literatures and sitting in front of computer and doing something, and we'll get some sort of result, and talking about p-values. Three different p-values we used to talk about meta-analysis. While we are talking about meta-analysis, three different p-values we used to consider. So it's all about statistics. No. The thing is, I'm not going to discuss anything about statistics. Because for statistics, we are going to apply some software which is used to perform meta-analysis. So what are the steps when we supposed to perform meta-analysis? What is meta-analysis? How it can be performed? All these areas which I am going to discuss in the analysis of analysis. Right? So here, one more thing I want to tell you while talking about meta-analysis, people are doing your prospective studies, right? In prospective studies, whatever the variables which you are collecting in the individual patient, because I don't think like we will get the exact result in each and every article what you are collecting. So some articles I'll show you the example which I got different, 4 different results. Some article says there is an increase, some article says there is a decrease, some article says there is no difference. We can, yes. No, you need to get the all variables in all studies, then only we can able to include the study in meta-analysis. Common variables, based on your objective, it's very difficult to go with the broad objectives in meta-analysis. You must be very focused in your research, thereby we can perform meta-analysis. Otherwise it's difficult. If you are keeping so many objectives, you won't get values in all articles. Yes, so I'll show for instance. It's a quantitative approach for systematically combining results of previous research to arrive at conclusion about the body of research, the definition. And meta-analysis is most often used to assess the clinical effectiveness of healthcare interventions. It does this by combining the data to or more randomized control trial. Here I want to make a note, it's not only randomized control trial. Now we can go with observational studies also. The thing is again, it's matter of variables which we are collecting. Why we are sticking with randomized control trial? Of course, the book says like it's randomized control trial, but I've seen many meta-analysis works which they have carried out by using observational studies also. The reason for sticking with randomized control trial is only because of the data. So I can able to generate the data which contains control and test so easily I can able to, that's why I used to stick with randomized control trial. Then the results also. We can strongly tell whatever the result which we are concluding. If you are using a randomized control trial, we can tell it a little bit louder. Then, why meta-analysis? It is important to be familiar with the type of data. So again, you people are aware about the data which you studied in biostatistics and research methodology, dichotomous and continuous, which is nothing but a categorical variable and a continuous variable. Whatever the variable which you are getting in the number, which is called as continuous variable, and categorical variable, good, better, best, all these things. Yeah, we can consider both variables for meta-analysis. And meta-analysis of trials provides precise estimate of treatment effect, giving due weight to the size of the different studies included. Before talking about the size, anyway, in forthcoming slides, I am going to discuss about the effect size, which we are going to use to perform meta-analysis. And coming to the when meta-analysis can be performed. So these are the reasons. So the first one is quantify the treatment effect and their uncertainty. Then increase the power of the results which we are expecting, and increase the precision. Explore difference between studies and settle controversies from the conflicting studies. Yeah. So this is quite interesting, conflicting studies. While I am searching literature, it doesn't mean that I get always positive report. Like if I am doing some intervention, okay. So the outcome may be always positive. It's difficult to get the positive result. Sometimes some articles may say negative, some articles no effect, and some articles will say negative effect. So we need to consider which one is, because we are looking for evidence-based information, which is very accurate. Like if you are, in case of economic or humanistic, we are not that much important, we won't give that much importance to the accuracy of result. But in case of clinical, we need to give importance for the accuracy. If, see for example, if I'm going to use one example as a biomarker. You're planning to do one meta-analysis in biomarker, which you need to very specific to article, even minimum to n number of articles. So what are the steps to be followed for conducting meta-analysis? The first one is research question, and second one is study selection, third one is data sources, fourth one is data abstraction, and fifth one is statistical analysis. So coming to the research question, you people are familiar with what is research question. From the research question, we are trying to establish truth, and like it starts once you plan to carry out this clinical research. It starts with research question, then study plan, then actual study. Is there any difference between study plan and actual study? Anybody? I think you people start. Just raise your hand. Just raise your hand. Come on. Then all are sleeping. Right. So there is a huge difference between study plan and actual study, because I just want to go with your own experience. You people are doing before after study. Before study, we are including n number of patients. Review patients. Yeah, of course, by submitting thesis that is different. But actually, while we are doing intervention, how many patients they are actually coming for review? Whether they are coming for review? If you are including 50 patients, hardly we used to get 10 patients per review. If you are doing, before after, I am planning before after study. So before I included 50 patients, but actually people returning to clinic and reporting to pharmacists only 10. That is the actual study. So I go with the research question. What is my research question and how I perform meta-analysis? Not I, we. Yeah. So coming to the neurotrophic hypothesis. This is the hypothesis which we have taken theoretically. Neurotrophic hypothesis proposes that depression is associated with reduced brain-derived neurotrophic factor levels, and that antidepressant treatment allows depressive behavior and increased BDNF level. In very simple, like antidepressant therapy which increases the BDNF level. So in case of major depressive disorder patients, the BDNF level will be low, right? So in normal patients, it's in normal state. So there will be an increase, no. It will be in the normal state, and in major depressive disorder patients, this BDNF level will go up. So in major depressive disorder patients, you are treating them with antidepressants and you are measuring the BDNF level whether it is in the normal or not. So if it is normal, then definitely we can conclude that BDNF and metabolic syndrome, BDNF diabetes, they establish a theory. So here whether BDNF can be considered as a marker in the therapeutic outcome. After treating these patients with antidepressants, is it worth to measure the BDNF level in those patients? This is what we want to identify. Okay, so we perform search. So search, and we used all search engines. From the search engines, you can see we got first 566 articles. Then next step, duplication. After that, removing the duplication, we got 571. Then again 66 article. One man can do reading all. There are some techniques which we are using to go through these all 566 articles. It's called FAST method, right? So finding, you need to consider the F stands for finding, and A stands for appraisal, and S stands for synthesis, and T stands for transferability. So in certain articles, it's very difficult. They may give in the graphical representation. Yeah, everything is good. Like your finding is correct, and your appraisal, they've done it in the proper study subjects which you want to estimate. Everything will be fine. Synthesis, you can able to generate the data. But while you are looking for the BDNF level, it will be available as a graphical representation, where the transferability is quite difficult. Either we should write to the particular author, so and so, we are working in this particular topic, we are in the need of this level, then sometimes chances are there you may get it, right? So all these things which we need to consider for clear-cut idea. Once, if it is based on this FAST method, you are bringing it to 14 articles. Once you got the selected article, you need to verify those articles by using PACO question. PACO model is helpful to outcome. So based on this, we need to work, and the questions which we need to address whenever we are searching. Once you finalize your area of research, your research question is ready, now you are ready with your article. So whenever you are starting this, you need to answer for either these are the questions which you need to keep in your mind. So how did you describe a group of patients? What are the most important characteristics of patients? And what main intervention you considering, and what do you want to do with these patients? And what is the main alternative to be considered, and what is the main alternative to compare with the intervention? It's not related to my example, I'm just giving, these are the questions which you need to consider while you are going for meta-analysis, right? Yes, so the very interesting part, data abstraction. Yeah, as I told earlier, sometimes you may get positive results, you may get negative results, right? So how to deal with these articles. So I just want to give you, based on the search, which here we have given search words like BDNF and major depressive disorder. These are the search words, and Hamilton rating scale for depression. So this is the quality assessment questionnaire which is used in major depressive disorder. These are the three questionnaires which we have given. while we started searching the literature. So after abstraction, we found only 6 articles that are suitable for our research. Coming to the next article, again here also, there was no significant difference in baseline and 6 weeks BDNF level between responders and non-responders. See here, there are 2 studies which we found like that, responders and non-responders. Whenever they are publishing a paper, they used to write, so based on, see here they used 2 things. One, they measured BDNF level, another one is quality of life questionnaire. Based on the quality of life questionnaire, they categorized patients into responders and non-responders. Ultimately, they measured the BDNF level in both groups. So that's why we have included in our study, right? So here they concluded like responders and non-responders. Like here, ultimately they are telling there is no difference between responders and non-responders, means there is no significant difference between the control and test, right? So the next article is an interesting article. This study was carried out in German, and these people are telling that after treating patients with antidepressants, there is a 12% decrease in the BDNF level. So I just want to show you the results of meta-analysis. So this is the forest plot which we used to get if we are applying the data in comprehensive meta-analysis. There are so many comprehensive meta-analysis since they are giving its free version. That too only for previously it was 1 month, n number of openings, and now they have reduced to 10 days. 10 days with 10 openings. That's it. So if you want to purchase, I think it costs around 1 lakh something. That too for 1 year subscription. If you want to have this meta-analysis package, particularly comprehensive meta-analysis. And the recent advance in comprehensive meta-analysis is previously we used to work with version 2, now they introduced version 3. Version 3 is user friendly and compared to user. Few things like what is forest plot? What we are going to conclude by visually what we can able to conclude by seeing the forest plot. This is what I am going to discuss. The squares shows the individual study effects, and the ultimate effect of all these works can be shown in the diamond. So ultimately we are looking for this p-value. This is the p-value which we are looking. As I told earlier, we are going to study design, effect size to assess the consistency of the effect across the studies and compute the summary effect. The solid squares are used to depict each of the study vary in size, and the vertical line in the middle is where the treatment and control have the same effect. The summary effect represented by diamond, and the I square is proportion of observed variation is real. The thing is, why we need to consider I square? Very simple answer. Why we started meta-analysis? Meta-analysis and meta-regression analysis is different. Okay, to perform meta-regression analysis, the I square value must be high. It's more than see here I mentioned mild, moderate, high. Mild is 25% and moderate is 50%, and the highest must be above 73%. So based on that quantity, we can go for further research. Then Q statistics and p-values serve as a test of significance. So as I told earlier, the 3 p-values, this p-value indicates here p is greater than 0.05. Ultimately, we are trying to say from the meta-analysis, BDNF cannot be considered as a marker to measure the therapeutic outcome. This is what we want to find out by using this forest plot. Right? So here, this p-value, if it is p is less than 0.05, definitely there is a difference, there is a significant impact of antidepressant therapy which increases the BDNF level. So here, since we got p is greater than 0.05, there is no significant difference between the group which we have compared. Right? So this is another forest plot where you can see the p-value 0.0 here with respect to BDNF and antidepressant therapy. And this meta-analysis study indicates the BDNF levels in patients with major depressive disorder may not be used as a suitable marker for predicting the efficacy of antidepressant treatment. Further studies we need, there are certain limitations. I just want to tell you, the duration of treatment while patients treated with antidepressants, some group they measured 6 weeks, in some other group they measured for 6 months. So there are limitations, but you must be very focused in your research question. Here we want to focus only on BDNF and its concentration and antidepressant therapy. That's it. Okay, so I'm really happy. This is my team. We work together. One among you, these 4 people, Vinny, Tanya, Nisha, and Sagar. They only work this work and take-home message. I'm not going to give any take-home message. So the thing is, you can see life is full of trials, right? So try to learn something new every day and enjoy the moment. Yes. There are 2 things, I square and P square. Okay. So here, that's why that's why I told, like, when we started with I square, what for we are using I square, we don't know, right? So after that only, why? Because we started with most of the published articles. So whatever the data which they have given, we try to find out those articles, those values. But now the T square value is there, which shows the heterogeneity. Heterogeneity, if you want to measure the heterogeneity, we need to go with this, yes.
After I square and the T square value, if you are using that meta-regression analysis.
No. The thing is, we need to consider the I square value for proceed with meta-regression analysis. If it is high, we used to go for, ultimately, it ends with P value. Whatever, even if you are going for meta-regression analysis also, it will end with P value. Based on that, we can able to find out the significance. The thing is, we will get the precise result. That's why we used to go for meta-regression analysis. Meta-regression analysis to know about subgroup analysis. Of course. Subgroup analysis. For subgroup analysis, we can go for meta-regression analysis, which we are considering I square value first. If it is high, then we can think of subgrouping. Like for example, in my study, if I want to
high BDNF and low
BDNF, like male, female, அந்த மாதிரி கூட கம்பேர் அந்த மாதிரி பண்ணனும். Yes, based on the I square value.
Then in meta-regression analysis, we use the R square value as usual.
Yes.
Okay, sir.
Any questions? Thank you. Thank you, sir.
Thank you, sir. Now we head towards the final session of the day. Topic